Okay, so in the previous video we looked at how calcium ions play a very important role in regulating muscle contraction. Where exactly do these calcium ions come from to begin with? Let's first look at the source of calcium in skeletal muscles, and then we'll look at how it's different in smooth and cardiac muscles. So if you look at the very general structure of a skeletal muscle cell, we have a sarcolemma. Basically, that's another word for the plasma membrane, covering all of the skeletal muscle cell. And it's important to remember that it also has these invaginations called transverse tubules. Transverse is just another word for perpendicular, and it's actually called that because it would be perpendicular to the filaments we see in the actual skeletal muscle cell right here. So just imagine that's the direction which the muscle, the myofilaments are, and this is perpendicular, and thus is called transverse tubules. And this is important because if you remember from our stimulation video, let's say we have a neuromuscular junction right here. This is a very bad representation of it, but this is just to give you an idea of the fact that there's a neuron synapsing at the motor end plate at the skeletal muscle right here, and you have the subneural clefts and all that, and then it causes the opening of sodium ion channels to cause depolarization and eventually an action potential, right? So this action potential is propagating from here. It propagates all the way down the T tubule, and in this way, this action potential is brought in all the way from the surface of the skeletal muscle cell down to the middle, or like deeper into the skeletal muscle cell, and that's very important to spread this action potential. And another important structure to remember here is the sarcoplasmic reticulum, which basically is in roughly this sort of a pattern right next to the T tubules. If we zoom into this, let's say this is the T tubule, and this is the rest of the sarcolemma. This T tubule, remember, is also covered by sarcolemma, so this is a continuation of the extracellular fluid down here, all the way down here. So we have the sarcolemma here, and if we zoom into it, it would look something like this. Remember, this is just the endoplasmic reticulum in muscle cells. And then we also have it over here, so on and so forth. And now we can focus attention on the receptor or the channels that line the T tubule. These are called DHP receptors, or dihydropyridine receptors. And they're in very close proximity to these sarcoplasmic reticulum ends, which actually have a certain name called the terminal cisternae. And you can see that here you have two terminal cisternae and one T tubule, and together they form what's called the triad. Okay, so we have these DHP receptors in close proximity with the terminal cisternae, and these terminal cisternae in turn have another channel called, let's do it here, the ryanodine receptors. In skeletal muscles, actually, they're so close to each other that these DHP receptors can mechanically change the conformation of the ryanodine receptors. And the way this occurs is, once you have the action potential propagating down the T tubule, it opens up these DHP receptors, which are actually voltage-gated calcium channels. And sometimes they're also called L-type calcium channels. So since they're voltage gated, it's important that the action potential travels down the transverse tubules, opens these DHP receptors, and changes the conformation of the channels that have mechanical, physical connection with these ryanodine receptors on the terminal cisternae of the sarcoplasmic reticulum, and in turn open these ryanodine receptors, which are also another type of calcium channels. But what they do is, since this sarcoplasmic reticulum has a high amount of calcium storage, calcium ion storage inside of it, once these ryanodine receptor calcium channels open, the high concentration of calcium ions inside the sarcoplasmic reticulum comes out, and so the calcium ions from the inside come out through these channels into the sarcoplasm. Let's say this is sarcoplasm, and this is the myofilaments that make up the contractile unit. And like we saw in the previous videos, these calcium ions then regulate contraction by interacting with these myofilaments. One more thing to add is that while cardiac muscle cells rely on this extracellular calcium ion concentration coming into the cardiac muscle cell to, in the end, regulate contraction, it's not necessary for the extracellular calcium ions to come into a skeletal muscle cell, because they've done experiments where depletion of all the calcium ions from the extracellular fluid of skeletal muscle cells does not affect this high concentration of calcium ions inside the cell to regulate muscle contraction. So it's important to keep in mind that extracellular fluid calcium ions are not as important to regulate muscle contraction as in skeletal muscles as in cardiac muscle. Okay, so now that we know the sources of calcium in the skeletal muscle, let's look at how they come into proximity with the myofilaments in the cardiac muscle cells. So here we have the same setup that we saw in the skeletal muscles, right? We have the T-tubule, which is the invagination of the sarcolemma, with its DHP receptors right here in red. DHP receptors, and then the sarcoplasmic reticulum over here, and the terminal cisternae having these ryanodine receptors. So it's basically the same setup, except you can notice how while there was a physical connection with these DHP receptors and ryanodine receptors in the skeletal muscle cell, here they are far apart. And you can also see that this was a triad in the skeletal muscle cells, but in a lot of cases in the cardiac muscle cells, it's a dyad where there's one terminal cisternae to one T-tubule. Okay, so let's go back to the fact that they're far apart. So when we have the action potential propagating down the cardiac muscle cell, and we know that this action potential it occurs because of what's called the pacemaker in the cardiac muscle, which is another name for the SA node. It causes self-excitation that causes action potential through depolarization, and then this action potential propagates down the T-tubule and reaches these voltage-gated channels or DHP receptors, opens them up, and the calcium channels out here in the extracellular fluid comes through these DHP receptors. And now this calcium is inside the cell now. And now this calcium induces the opening of these ryanodine receptors, which then open up and allows the calcium from inside the sarcoplasmic reticulum come out of these ryanodine receptors into the sarcoplasm of the cardiac muscle cell and allow contraction to occur, just like we saw right here in the skeletal muscle cell. Now, the reason why these calcium ions go from the extracellular fluid to the inside is because there's a high concentration of calcium ions outside the cardiac muscle cell. A key mnemonic to remember is actually the salty sea surrounds us. So if you remember that, that means salt is sodium and chloride, and the sea in this case would be calcium. So it surrounds us, meaning it's outside the cell in a high concentration. And here we're focusing on the calcium ions. They would rush in through the open DHP receptors and in turn induces the ryanodine receptors to open, allowing the high concentration of calcium ions from the sarcoplasmic reticulum to then come out to the sarcoplasm and regulate contraction. So this is actually called calcium-induced calcium release from the sarcoplasmic reticulum, or CICR. Okay.